I present you KÅRE MØLBAK, which is currently vice president of the Staten Institute in Denmark. The Staten Institute, in fact, you can see it at a mix of something like the scientific France in VS, but with more neurons, more laboratories, and a way of making vaccine immunization. And it will present you then the ways of collecting the data for doing information surveillance, doing surveillance in Denmark.
Okay, thank you, and it's a pleasure being here in Marseille. So this is my first time in Marseille, and I love it. So I hope I can come back another time. I'll talk about how we integrate things in Denmark, what do I mean by integration? There are two dimensions of integration, where the first one is about integration of microbiology and epidemiology using information technology, and then there's another type of integration, which is the One Health integration, where we try to integrate human and veterinary medicine and other branches of sciences like food science, and then of course the environment and climate, which becomes more and more urgent. So that's really the two dimensions of the integration that I would like to talk about. But I'll start by giving a small introduction about healthcare in Denmark and my institute. I'll talk about how we use whole genome sequencing as a routine tool in surveillance. And I'm not a very technical person on this matter. I'm an epidemiologist by training. So if you have any detailed technical question, I must refer to the people back home. And you're welcome to contact me afterwards. Also, you can have the presentation with my email address, so you can contact me if there are any questions. Then I'll talk about the One Health integration, and then I'll talk about the integration for information technology and how we can use our data in a real-time way for making decisions. And then I'll talk about some of the challenges we have and perspectives. So Denmark is a small country in the north with a population of only 5.7 million people, and the geography is a little bit peculiar with this peninsula that is connected to Germany, and otherwise it's just a bunch of islands. So it's divided into 5 administrative regions that runs the hospitals. And most of these are publicly. There are only very few private hospitals. They are highly specialized, and then we have 98 municipalities, and they are responsible for primary health care and also the child care and the care that takes place in schools and so on, school nurses. But the general practitioners, they're actually private in Denmark. But all the costs are reimbursed, so it's free to go to your family doctor because all the expenses will be reimbursed by the regions, and then they are also very heavily regulated. Otherwise, this will not work. All antibiotics is per prescription. And the microbiological services is served by only 10 departments of clinical microbiology that are publicly owned and located in the hospitals and are connected together by the internet in a secure way. And then we have the individual person number that is used for all healthcare services, which is the key to link all the data together. And I'll come back to that. A little bit about my institute, which is funded in 1902 as a facility to produce diphtheria and tetanus serum. So that's why it's called the Staten Serum Institute. So it was a group of people, entrepreneurs, that broke out of the University of Copenhagen and wanted to make production of anti-serum, and they continued to develop microbiology, to start vaccine production, And this is the line outside the institute in 1941, where people are queuing for diphtheria vaccination. So the issue about vaccine hesitancy was not a big concern at that time. People knew that diphtheria was a serious disease and was eager to get a vaccine. And I also know that in France, vaccine hesitancy is becoming a challenge because people forget about the diseases. They don't consider measles, rougeole as a serious disease anymore. But these were different times. So you can say that the institute developed really in the tradition of Pasteur, where you have microbiology and applied research going hand in hand with public health. So we do clinical microbiology and public health microbiology. So we have still some diagnostic functions left, but then we also serve as a reference laboratory. And then we are responsible for a lot of the preparedness, where we support the National Health Authority with doing outbreak investigation and biosecurity. So we have a Center for Biosecurity and Biosafety. We do the public health surveillance. We do training, both in specialists in public health and in clinical microbiology and otherwise. We have a lot of international collaboration, and then we are in a very exciting moment now at our institute, because we are now also being responsible for the veterinary contingency diagnostic and preparedness counseling and advice within the veterinary area. So right now we are building a laboratory for swine and mouth disease, and for swine fever and foot and mouth disease, rabies, avian influenza, all the highly infectious animal diseases, which is a great opportunity really to practice One Health. And this is done together with the University of Copenhagen, the veterinary school. And I happen actually, in addition to be vice president of the SSI, also to have a part-time professorship at the veterinary school in Copenhagen. So all this happened actually in 2017 when the vaccine production was sold. So the vaccine production that used to be part of our institute was sold to a Malaysian company called AJ Vaccines. So we are now down to about 700 employees at the institute. It used to be around 1,200. Then we had an internal reorganization, and I became vice president, and we decided to integrate the Department of Epidemiology with the reference laboratories, which means that we have assembled the wet and the dry sciences under one umbrella. And then we have been fighting for years with the regions about which kind of laboratory activities should be placed in the regions, and which part should be placed at the SSI. And we made a peace agreement where we have a council now that decides which activities to run at my institute and which activities to run in the regions. So we get paid by the region, and then we decide together how to use this facility. So we're not paid sample by sample, but we receive a lump sum of 150 million Danish kroner, which is about 20 million euro, and for that we can do reference activities and diagnostic activities, which means that they for free can send samples of, like if they have an outbreak, hospital outbreak, they can send the samples, and we can do whole genome sequencing for free, and we can run national diagnostics for tuberculosis, because this is a very specialized area, and so on. And also many of the emerging diseases we have centralized diagnostic at SSI. And then as I said, we won this tender for the veterinary preparedness, and we are building this now, this dedicated high containment laboratory, which means that we can have a One Health approach to the veterinary service. So we have now much better surveillance of the zoonotic agents. So like we have one laboratory that do salmonella, we have one laboratory that do influenza, independent on whether the isolates come from swine or birds or wild birds or humans. So that's a big advantage, and the possibility of also doing comparative research, use animals as models for human infections. And then there's also many management advantages of having the labs together. And this is our organization where we have my division, which is we have the infectious disease immunology and prevention, bacteria, parasites and fungi, virus and microbiological special diagnostic, and the International Reference Laboratory of Microbiology. And all in all, it's about 300 people work in this division. Then we have a division of for doing surveillance of congenital disorders, and they also host the Danish National Biobank, which is a biobank where we have about 30 million samples from the Danish population taken in various time, and it's a repository that anybody can access. Then we have research unit for vaccine development and for epidemiology. A little bit about how we have put up our system for the use of whole genome sequencing. We have set up a system with a central facility for DNA extraction, for the sequencing, for the quality control, and then it can goes into several pipelines when it has passed. And we use the data for outbreak detection and investigation. We use it for the antimicrobial resistance surveillance. We use it for reporting of data to the European surveillance system, for WHO, and for scientific studies, and for presenting at our websites. And then external partner can also access the data if we do it for research purpose. And there are several workflows. So we have the main workflow, which is run on a NextSec platform that is run 3 to 4 times per week. And then we have a special setup for microbiome analysis, which is done around twice a week. And then we have a third way, which is the fast track, where we do the manual purification and library preparation, and where we run it on ad hoc basis on an ISEC 100 platform. And for all these, we have automatic transfer to a server and runs through the common quality control pipeline, and then a notification will be sent to the responsible expert when the data are ready for analysis. And the current flow is about expected at 10,000 for the whole genome sequencing and 3,000 microbiome analysis. And that's something that we expect will increase in the coming years. So this is the staff of bioinformatics at the KÅRE facility. So you can see these are young and nerdy people who work, and then they support these expert areas. So these are the old microbiologists sitting there and using the data from the young bioinformatics. And we have developed this quality control pipeline that is called Bifrost. So this is an example of an output from a Clostridium sequencing. This is from a Shigella sequencing. This is the checker of the quality control room, where you can see there's one that has failed and these data will be available to all on a platform. So I'm sorry, this is in Danish, but this is a little bit about how it's organized. So a patient on a sample a patient on a hospital will take a sample that is sent to SSI, or it can be sent to a local department for whole genome sequencing. So some of the larger laboratories in Denmark also do whole genome sequencing, and they use the same quality control pipeline, and then the data can be stored in their local folder, where it's only the local department that have full access to the data, or it can be stored on the national data folder. So for example, if it's an MRSA, it's something that will form part of the national surveillance, so it will go to the national folder, where both the local department and SSI can have access, and then we have also our own SSI data folder at this server that we can access for our own purpose. And again, if it's for part of the national surveillance, it will go to the national data folder. And it's all stored on a high-performance computer at the Danish Technical University. So the whole storage has been outsourced to the Technical University that has this supercomputer. And the advantage is that all the isolates are run through the same quality control pipeline, independent of whether it's a private research project that is sort of owned and managed by a local laboratory, or whether it's part of the national surveillance. And the data that has been analyzed as a part of research project can then also later be moved to the national folder and then form part of the surveillance if that's needed. So we have a national quality control and sharing of the data. Then we have developed a fast track flow that is quite interesting, and this is how it works for the carbapenem resistant organisms, which is one of the concerns that we have. So again, we have the patient where we do the culture, and we do the phenotyping, and then if it's shown to be a carbapenem, the isolates will be sent to our institute, and we actually have a car that starts around midnight in the northern part of Denmark, and then it passes through the 5 regions and ends at my institute in the morning, which means that a sample that has been processed during the day can be at the institute at 8 o'clock in the morning, and then if the isolate is flagged for the fast track whole genome sequencing, it can go into this fast track pipeline with DNA purification at 10 o'clock, library at 2 o'clock, and then it will be run typically in the ISEC 100, and the next day there will be results available, and the bioinformaticians will meet early in the morning and have a core genome MLST at 8 o'clock. Then SNP analysis will have to wait until later on during the day. But that means that if there, for example, an outbreak in the hospital, they'll have data in a very timely manner. Then, of course, we have been thinking a lot about how to organize this, and we have decided on outsourcing of the storage to have this priority of a risk clone that can go into the fast track typing, and we have a core facility of bioinformaticians, and currently we do sequencing at SSI and Hvidovre and Rigshospitalet And then they have at some other hospitals, they are coming up as doing that, and we work together in a network to share the data. Now this is an example of how we do it for contact tracing of carbapenem resistant agents, and we have developed a tool which is so this represents an outbreak, and these are the dates, and these are different patients, and they are tracked on their course of admissions. So these different codes represent different wards. So this is done by integration of administrative data from the patient registry with data on the typing of the bacteria. So the trick is that we identify an outbreak by whole genome sequencing, and then we integrate these data with administrative data from the hospital discharge registry, where we use a software that we have developed that is called EpiLinks, which automatically goes into the administrative data, and then it tells where the patients have been in the hospital when the isolates were taken. And by that we can identify that the most likely source of this clone was this department, which was found for 17 of the patients. And then we also start to use the Minion for doing fast track typing. This is a little bit about the priority organism. So it all started with the foodborne agents back in 2013. So the gray are the sort of the period where we plan to use whole genome sequencing, and then we have evaluation, and then it started as a routine for Listeria and Salmonella, ESBL producing organisms, and vancomycin resistant enterococci, which is a big concern we have, and Haemophilus. And then the STEC came later on, Campylobacter, meningococci, Legionella, and Clostridium difficile. And MRSA came later on, group A, Streptococci, and also for virus and tuberculosis. So this is sort of has been the sequence where we have been adapted, and now it's replacing a lot of the phenotypic methods. And these are examples of outbreaks of Salmonella, where we have been using whole genome sequencing, and so this one was identified as with this sausage as the vehicle, and we also do it for these are STECs, and we use it for identification of clones, and in particular this is also used for doing differentiated risk management based on the genotypes. And then we do it for in silico surveillance of resistance, and I was proud to read this editorial in Eurosurveillance that whole genome sequence data enable comparisons between countries with an unprecedented granularity. The Danish strategy to perform whole genome sequencing on resistant strains is an example how microbiological surveillance ideally should be performed in this area of antimicrobial resistance. So we were able to go back into the databases and identified colistin resistance in Enterobacteriaceae. So this was a little bit about whole genome sequences. Now I'll continue my talk on the One Health aspects. And actually, we have a long tradition in Denmark of working together between the sectors, and Salmonella is a very good story where we see the emergence of Salmonella in Denmark that came into several waves from broilers, from pork, and from eggs. And in 2008, we had a major outbreak from pork, What we did was that when we saw this emergence of Salmonella, that we created a zoonotic disease response group and a zoonosis center, which both have groups where authorities and research surveillance institutions work together, but we also do have a link to the stakeholder groups. So we meet with the industry, we meet with the consumers and their organization, and we have implemented a number of actions plans that has a lot of focus on the pre-harvest measures, sharing of methods and data, and we have a joint reporting called the annual report on zoonosis, and we have a joint action group that meet every week to talk about the current outbreak. So it's important, in my mind, to have a system where people meet also in times of peace, because then when you have the crisis, it's much more easier to manage. So this is the annual report on zoonosis, and this is the Danish Salmonella account, where we use typing of bacteria in combination with epidemiological data to track the different sources of Salmonella. And we also do that for assessing the outbreak. So this is the 64 outbreaks that we had in 2008 that were national and that were investigated. Then also for the AMR field, we have a long tradition of working together. So we hosted actually back in '98 the microbial threat conference, and if you read the report from that conference, it's interesting to read, because it's actually also what is written in many of the declarations and the minutes of meetings that you see today, that we need to work together and act. And we developed the Denmark surveillance system, where we have a joint monitoring of resistance and antimicrobial drug consumption, both in humans and in animals, providing data for risk management. And this model has become a model for many other countries, as you see here. Now, one of the examples of One Health collaboration is the work that we done around Q fever. And because I'm here, I wanted to just present 2 slides about Q fever. Because actually prior to 2003, it was not considered an issue in Denmark. But then in 2003, there were analysis of bulk milk from dairy cattle that shows that Q fever actually was widespread in cattle. So there was serological evidence of Q fever in the cattle farms, and about 40% of the bulk milk samples were seropositive. And that caused concerns in Denmark, in particular because nowadays a lot of the veterinarians, they are women, and they may be exposed to Coxiella when they work at the farm's assistant in delivering birth and so on. We saw an increase in testing. So in 2006-07, there were 1,630 individuals tested for Q fever, mainly those that were in contact with livestock animals or were with veterinarians. 11% were positive, and there was also a survey done, published in 2010, which again showed 11% positive, and it was highest among veterinarians, 36% positive. And in particular for those who had contact with cattle, and that caused some concern in the industry, and we managed to set up a research project that was the PhD thesis of Stine Yde Nielsen, who took advantage of the Danish National Birth Cohort. So the Danish National Birth Cohort was a study where we follow up on about 100,000 Danish pregnancies. And this is actually a resource that is available also if you want to do studies. Then you can, then it's all documented in English, and there's also biological samples that are available. So right now they are doing follow-up of the outcomes of those 100,000 pregnancies. Now they are for the 18 years survey, and some of these women, they have now grandchildren, but we still continue to follow up on those pregnancies. So there are samples taken throughout the pregnancy and then at the offspring later on, and there are interviews at regular time. So there were 856 of the 100,000 pregnancies included in that PhD study, and of those who had livestock exposure, it was 47% that were positive for Q fever in the ELISA, and then confirmed by immunofluorescence, mainly phase 2 positivity. And for those who had hobby animals, animals at their private farm building, but not doing big-scale commercial farming, it was 30% positive, and those that were unexposed to farm animals, it was 4.8% positive. But then we had data from this cohort where we looked at the risk of miscarriage amongst those that were seropositive, and it was an odds ratio of 1.5, and risk of preterm birth was 0.4, so we could not really document any risk for the offspring in that study. And also 14 of the 100,000 women compared by seroconverted by ELISA standards, so they were ELISA negative at the first sample, and then they had IgM at the second sample, and they were also confirmed. 10 of these 14 were converted by immunofluorescence, and all gave birth to live babies at term. So that was a bit reassuring, and I mean, this is something that we can discuss, but it could be related to that these are exposed from cattle and with a very low degree of exposure, where the situation could be different if you have Q fever in goats or perhaps also sheep, and a higher degree of exposure. So it's probably a different strain of Coxiella in Denmark. And now the situation is very common. We see a few cases now and then, but it's something that we can count on one hand per year. And it's very rarely that we see cases where it actually affects pregnancy, but it happens now and then. But it's not a big public health concern. Now, another issue that we have been studying quite closely is the livestock-associated MRSA, and as you may know, Denmark is an important pork producer. So we have a population of 5.7 million people, and we have a production of 30 million pigs per year. So it's about 6 pigs per person in Denmark in average. So it's a big industry and very vital for our economy. And what we have seen since 2010 is this increase. So this is the number positive swine herds. So it's now up to 90% positive herds that have MRSA, which is this clonal complex 398. So it is spread within the pork industry, and then the farmer get exposed, and also it can be spread in the environment and to families. And of course, the concern is if it is spread further into the community and to hospitals. So it's an issue that we follow quite closely. And this shows the emergence of different types of MRSA. So the green line here is the number of infections with the livestock-associated MRSA. And this big increase we see here is due to a screening practice that was established in 2012, where exposure to animals were identified as a risk factor, along with other risk factors. So this is like a surveillance artifact, and that's also why it has been stabilizing now, that now all farmers are being screened whenever they are admitted to a hospital for MRSA. And so these are the healthcare-associated MRSA, which is a very low and stable level. And the blue line here are imported community-associated MRSA, which also on the rise due to more and more travel and the occurrence of MRSA in other countries. And we have made research on how to manage this issue, and the issue is now with a 90% positive in the farm animals. It will be extremely expensive to eradicate the problem. It will be too costly for the industry. So our management practice is that husdyr MRSA blive I stalden, which means that let the animal, the livestock-associated MRSA, stay in the stable. So with hygiene, we want to keep it in the stable. So have increased biosecurity around the stable. So all the stables, pigsties in Denmark, should be equipped with a washing facility, so that you can shower when you go out, change your boots, change your clothing to let the MRSA stay in the stable. We have studied the MRSA comparing clinical and livestock associated, and what we see is that it's exactly the same strain that we see in severe bloodstream infection as we see in the farm animals. Of course, there may be a little difference in some of the prophages that they contain or other virulence factor, but essentially it's the same strains that cause severe infections in humans as we see in the cattle, and we can also see that the expansion in farms is caused by 3 different clones, which we call L1 to L3. So a lot of the emergence in the swine industry is due to these 3 clones that contain additional determinants of drug resistance that enable them to survive in the pork environment, where there is currently used a lot of antibiotics, including tetracycline, but also zinc as a way to manage weaning diarrhea among the piglets. So these strains, they have also resistance against zinc. There's a lot of discussion now in the pork industry about what should the future be of the Danish pork industry, given that there will be more and more demands for the consumers of having a sustainable production. And this is an example how the food authority now can benchmark a single producer against the development goals of the UN in terms of the management, the land use, animal welfare, discharge, water use, energy and climate, biodiversity, occupational health, life, quality of life, and economy. So this represents a single farm and how is the quality of life if you work in that? How is the economy of the farm? How is it managed? What is the land use? What is the animal welfare? What is the discharge? So in terms of discharge, this farm is not doing well. The water use, energy and climate, again, there's room for improvement. So this is maybe an example of how agriculture will develop in the future if we really think about planetary health, that we should mark the performance of the farmer not only for economy but also against a broader range of measures. I think that's quite thought-provoking. And also we have a new trend that is called opdrættet uden antibiotika, that is raised without antibiotics. And that's also becoming a trend. So the pig should receive no antibiotics from birth to slaughter. They should have been fed with vegetable-based feed, and they are tagged with a special raised without antibiotics tag, which are being removed if they are treated, and there is an annual independent production audit. So the Danish food authorities are now really working together with the pork business to develop new ways of sustaining this business. Okay. Now a little bit about data for decision making. Now the e-health in Denmark, I'm totally shifting gear now from going from farms back to hospitals and primary care. So it's we have a lot of different IT systems. So the private doctors, they have their own system. We have the 5 regions, and they have also different systems. And then there are again a whole range of systems that are being used for IT in nursing homes, a public health system, and so on. But the trick is that we have common standards for communication, data exchange, and reimbursement, which means that there is a national protocol if a doctor sent a prescription to the pharmacy, or if the doctor sent a lab request to the local laboratory, and when the report from the laboratory is coming back into the clinical department or the office, and also communication with the national registries. And we can take advantage of these protocols. So you can see the facilitators for developing e-health has been 2 issues, that we have this unique personal identifier that allocates to all citizens at birth, which means that every sample can be linked to a course of admission, it can be linked to a vaccine registry, it can even be linked to data on the cost of treatment or outcomes, whether you are returning to the workforce or you are continuing to stay on public income support. And then we have a legislation that actually allows data linkage and actually encourage that we use the data. So that's what you're going to talk with your lawyers about, is that if you could make a change in their mindset, that we actually would like to use this data for the public good. Of course, there are some demands, and with a GDPR, there is an increased demand of that you make your case very clear that you want to use this data for A, B, and C, which I think in many ways is good. So there's an increasing demand for specification of the need you have. But if you do it for a good purpose, then you're allowed to do it. The most important step that we took was that we developed the National Microbiology Database, which is a national real-time database that have copies of all reports that are produced in Denmark, whether they are positive or negative, which means that my institute can have a total overview of all testing done in Denmark. And this has really been a collaborative effort of all the microbiology lab and the Department of Infectious Disease and the IT system providers. So we have been sitting together working out this system. So it's a network that has been that also has increased the collaboration between the Danish microbiologists and those who work with the surveillance center. And it's also been a continuous development of the e-health system. And it started as a proof of concept in the capital region, where some of the microbiology department wanted to share their results. When patients were transferred from one department to another, they wanted to see the results produced by other departments. Then they went to me and said, why don't we make this a reality on the national scale? So it was accepted by the Minister of Health as a national project, and then 2 years later it was launched. So it's national sharing, it's for the national surveillance, and it's a data source for healthcare projects, research and quality assessment. And it has really become a primary source of having access to microbiology results. So when patients are being transferred from one hospital to another, they would have on admission a full overview of all testing done on that patient, which means that they can save time and save sample, because they know that a culture was taken at another department, and they can access to the result of those data. So whenever a sample is being sent to a hospital, the test report, a copy of the test result is going back to MIBA in real time. So we have the same overview as the doctor who sent the samples, and we can extract those data in real time. And we have what's called a central dynamic mapping. So there may be a different way that they use this standard exchange form, but we do a national mapping, so we can extract data for surveillance purposes. So it's complete data at a national level, both positive and negative test results. It's real time, and they are personalized, containing this civil registry number, so it can be linked to causes of admission, drug use, and so on, which essentially it's like a big cohort study where you can follow the entire nation, and there's no extra burden of reporting. Now, one of the limitations has been that it was originally just a copy of the result that was sent to the clinician. So this could be a urine sample that has been sent to for culture and phenotyping of drug resistance. It's an E. coli that is resistant to ampicillin, but sensitive to sulfa and gentamicin. But of course, this sample has been tested for others, so we don't know which other indicator drugs were part of that. They probably test for 10 or 11 different drugs, but this only showed 3, and we don't see the zones, we don't see any MICs, and there's no other information. So this missing data are now being included in the next generation of the support. So we have a lot of additional data coming in that is not shown to the clinician, but can be used for surveillance purposes. And there's also a flag in these data if the patient is exempt from research purpose. So you can say in Denmark that I don't want to have my data part of research project. And that's also going along with these data, which means that we can see whether these data can be used for a research project or only for public health purposes. So there's a lot of detailed information that is coming along in the new version. And these are just a couple of examples how it improved our surveillance. So this is the reporting of neuroborreliosis in Denmark. And after we got data from MØLBAK, we had almost 3 times number. So you can see that we usually under-ascertain the incidence of neuroborreliosis infections in Denmark. And this is an example of how we use it for the influenza surveillance, where we have data from the National Patient Registry, where we know whether the patient were at hospital, whether the patient was admitted to intensive care, and whether the patient had chronic conditions beforehand. And we know the vaccine, if it was given, and we know from the civil registration system where the patient is living, and if the patient had died. And with this type of integration, we can make real-time assessment of vaccine efficacy. We can make a real-time assessment of severity, so we know exactly how many of the positive patients were admitted to intensive care unit, and how many died, which is a tremendous advantage when we want to assess severity of influenza. This is just data from MØLBAK, where we actually can see the testing practices. So these are the number of tests taken by season, which is increasing, and which is partly due to a lot of use of point-of-care testing now, which is established in most of the hospitals, and we can follow that development. So it was 29,000 samples in Denmark in the season 2016-17, and now it's up to 55,000, and we expect it to be even higher in the coming season. And we are using these data also for doing surveillance for healthcare-associated infection. So based on the date of sampling in relation to the course of admission, we can establish whether a sample, for example, of a bloodstream infection was taken in the time window 48 hours after admission or later on, which means that in mathematical algorithms we can make real-time surveillance of healthcare-associated infection. So this is currently being done for Clostridium difficile, for bacteremia, for urinary tract infection, and selected surgical site infections after hip and knee replacement surgery. So this is the wider integration of MØLBAK, where we link up with all the registries. I actually think I'll stop here now, because I did a lot of talking, and it's 5 to 1, and I touch upon a number of different issues, going from pig farming to integration of data and the use of whole genome sequencing. So perhaps there are a few questions and comments, and then I would be happy.